Go.
Hello, biology students of America. My name is Andrew, and today on BCTV, the main topic is cloning. Can we actually do it, or is it just scientists with crazy dreams? After about 20 minutes of research, we found out that the first cloned mammal was a sheep named Dolly. Dolly was cloned from a somatic cell from a dead adult sheep. This process is known as somatic cell nuclear transfer. For more information on this exciting procedure, let's go to Tyler in the laboratory. What Andrew just said is correct. They use this process of somatic cell nuclear transfer. To do this process, you first have to get a somatic cell, just like I have here, from a human or animal that you want to clone. Then you take the nucleus out of the donor egg cell and transfer it into the somatic cell. Once we do that, we just shock it with a jolt of electricity and fuses the two together. Simulating the egg causes it to divide just like it would with a sperm cell. A lacticized or an early stage embryo forms after about a week. An embryo transfer specialist transfers it to a recipient female where it comes to develop. After having a full-term pregnancy, the recipient gives birth to the identical twin of the genetic donor. Well, that was a very good explanation for our very own and very overpaid scientist, Tyler. But the question that we keep on getting are what good and bad will come from cloning. Now we go to a promoter of this new technology, Josh.
Thanks for having me on the show, Andrew.
No problem. So, what are some positive outcomes on cloning?
Well, one obvious outcome is that you can make human life. Say you have a dead family member, you can use cloning to recreate them, almost like bringing them back from the dead. This can also be a positive thing for homosexual couples. One of them can be cloned and can have a child of their own instead of adopting. Another way that cloning can help our society is by fixing defective genes. If we figure out how to change a person's gene, then we can replace their defective ones, almost like having interchangeable parts for your body. That idea brings us to the fact that we can replace human organs and tissues. We wouldn't have to make people wait for a new heart or lung.
Thank you, Josh, for that positive outcome on cloning. Our current researcher, Chase, says no this. He says the danger of failure outweighs the success rate. He now joins us from the studio. So, Chase, what are some negative consequences of cloning?
Well, with cloning, as everything else, there's a chance of failure. Only a small percent of embryos survive the process. Even if the embryo survives the process, it has a high chance of getting a genetic disease or dying later. Through the years of the clone, it can lose the diversity of its genes. They have a weakened power for adaptation, and that means that they will be affected by, or everyone will be affected by the same diseases, and we won't be able to cope with the changes in the environment.
Well, that was something new to hear. Well, you've all heard it here. I hope you've all learned something from our scientists and our experts. Thank you.